Hi everyone, welcome to the third video in the DNA replication, transcription, and translation series. We've already covered the structure of DNA, we've covered what replication is, and now we're going to start getting ready to talk about transcription and translation. But before we do that, we've got to talk about a new molecule that's very similar to DNA. It's called RNA, and I'm trying to get to that point, and to do that I got to tell you a little bit about what's going on. You see, DNA is not just there to copy itself. That would be stupid. It has to have more of a point than to just make a copy of itself. What does it actually do? Its function is to store genetic information, and that genetic information then gets copied and used to create something called a protein, and proteins make our traits. The reason you look like you and the reason I look like me is because we have certain proteins that make up our body structures, and by the way, those proteins do a lot more than just build us. They also influence our behavior. So our behavior and our body shape and appearance and how it functions is all governed by proteins, and to make those proteins, the information to do that is found on DNA. Now, getting that information from the DNA into a protein and have it express itself is a very complicated process called transcription and translation. It involves an intermediate molecule called RNA, and that RNA molecule copies a small portion of the DNA called a gene and takes that copy outside of the nucleus to where we can turn it into a protein. Let me talk to you about a gene for a minute. Here we have a picture. Again, we've looked at this a few times. We are talking about the chromosome here at the top, and we know that this is just a bunch of coiled up DNA. So what I'm really trying to show you here is that we're looking at the DNA, and the DNA has a lot of information on it. Now, there are blank sections of the DNA in between this sets of information where there's not really much, it's just gibberish. But where we have pieces of information that we use to make proteins, these are called genes. Now, I'm sure you've heard of this term before, but I want to be clear. A gene is a set of information, a set of this code, a slice of this code that is found on your DNA molecule. It is not both sides, it is only one side, and it is copied in a process called transcription. Now, that transcription happens using RNA. So this is all inside of the nucleus. There is a slice. This is actually very short. There's no way it's this short. Genes are actually probably thousands of these letters long, but just for sake of showing it to you, this is the best picture I could find. By the way, because this is a very long molecule and we need to know where does the gene start and stop. Every gene starts with a promoter and ends with a promoter. There is one start promoter, so every gene starts with ATG, okay, but sometimes I think there are 3 stops, and so you'll know when a gene starts by seeing ATG, but this tells us where to start copying the gene and when to stop, okay. Otherwise we would copy the whole thing all the way down and it creates a real mess. This leads us to the idea of the central dogma of DNA. And I want you to see this. It's a picture here at the bottom. It's also written right here. DNA contains the information, but through transcription we turn that into an mRNA molecule, which leaves the nucleus and goes outside to where it's turned into a protein. And then I added that the proteins make traits. So this, you may want to write this down. The central dogma is that DNA is turned into RNA, RNA is turned into proteins, and proteins make traits. Now, what it really means is that your traits ultimately come from information found on the DNA, but there is a pretty complex process in between the two, and we're going to be talking about that. Let's talk a little bit about what's going on here and why. Why don't we just go straight from the DNA to the protein? It's because of this. If you can imagine that we're building a house or something, we have a master plan, that's the DNA, but the master plan, we only got one copy of it, and we don't want to take our one copy out to the work site because it'll get, run over by a tractor or torn in half or something, who knows what. So instead, the nucleus makes a copy of the master plan and gives it to an intermediate, somebody like this lady here, who then takes it out to the construction workers. That'll be these guys here. This lady right here, she's the messenger RNA, or the mRNA. Her job is to take that copy, actually in this analogy that copy would be the messenger RNA, but we'll just put the two together. Her job is to take that message out to the workers, where they then complete the construction of the protein. Notice that we're only taking a copy. If something happens to that copy, no big deal, we can come back and get another copy. We do not take the actual master plan. That would be the DNA. That stays safely inside of the nucleus. Instead, we just make a copy of a very small part of it. In fact, we're not copying the whole thing. This isn't a very good picture, but we make a copy of a very small part of it, just the specific part we need to be working on today, and we carry it outside of the nucleus to where it gets done. That's the best analogy I can give you. Okay, the RNA is disposable. The RNA is just, once it's done, it can be torn apart and used again, and it's no big deal. Okay, so let's talk a little bit about the difference between DNA and RNA. DNA is a double strand, where RNA is only a single strand. Okay, DNA is usually very long. We're talking about billions of these letters, where RNA is only about thousands, maybe not even 1,000 in some cases. And DNA uses guanine, cytosine, adenine, and thymine, whereas RNA uses guanine, cytosine, adenine, and uracil. So there is no thymine inside of an RNA. Instead, wherever there should be a thymine, there's a uracil. Now you need to remember that rule. Okay, if you ever see RNA with a T, you know something's not right, because it's a trick. Don't fall for it. Okay, this is, other than that, this is just like DNA. It is a polymer made up of nucleotide monomers, and it behaves in exactly the same way, as I hope you'll see in a minute. And we copy the DNA, or that little piece of the DNA. Instead of copying the entire DNA, we're just copying a little piece called a gene. We copy that gene using the same base pairing rules as Chargaff showed us, like if we were trying to copy the entire strand, but only we're copying a small part of it. So if there's a G here, there's going to be a C over here. If there's a C in here, there's going to be a G there. If there's a T here, there's going to be an A there. And if there's an A here, there's going to be, don't say T, because it's actually a U for uracil. Okay, let's talk a little bit more about that. And this is just a cooler picture, but you know, the backbone that makes up deoxyribonucleic acid is made out of deoxyribose, which looks quite a bit like ribose, but you'll notice that right here, this is an OH, where that's only an H. Okay, this does not have as much oxygen as the ribose. Ribose stands for ribonucleic acid. It's the R in RNA. It's got an oxygen there. Okay, but over here, the deoxyribose, it's missing that oxygen. That's why it's called DNA. It's missing an oxygen. That's why we call it deoxy. Deoxyribose means it's missing an oxygen. And that's what this really is. So here we have this deoxyribose over here, and the whole thing's made out of that. But the RNA is made out of a slightly different nucleotide. It has a slightly different structure to it. It's got an extra oxygen, so we call that ribonucleic acid. Okay, the three bases that are common are cytosine, guanine, and adenine. You're only going to find thymine inside of DNA, and you're only going to find uracil inside of RNA. And here is another look at that master plan, the idea that the DNA stays inside of the nucleus. But as you can see, we open it up a little bit. We talked about those bubbles in video 1 or video 2, I forget. But we open up a little bit, and instead of copying the whole thing, we're just copying a little piece of it there, that gene, and it gives us a much shorter strand of a nucleic acid. And so this polymer is much shorter. It looks long, I know, but compared to this thing, it's very short. It's copied inside of the nucleus. That's called transcription. And then once the copy is made, it has to leave the nucleus and come outside to the cytoplasm. So it comes out through holes in the nuclear membrane called nuclear pores. It comes outside, and once it gets out, now we're going to do a second part called translation. The transcription is pretty easy. It looks a lot like replication, so it shouldn't be that hard to understand. The translation is a little bit harder, but they each have their own video, and we're not even going to go into that on this video. But so it's a two-step process. I make a copy of a gene in here, and that copy is the messenger RNA. That should have a little M in front of it. And that messenger RNA, like this, comes out, and step 2 is we take that information and we use it to make a protein. This is the goal. This chain of amino acids that goes together to make a very long structure called a protein, and that protein folds up to give us to build our bodies. And then, as it turns out, that's how we get every single thing about us. The reason your eyes are colored the way they are, the reason your skin is colored the way it is, the reason your nose is shaped the way it is, the reason your blood does what it does, etc. I mean, there's literally millions of traits, and it's all because of the proteins that you have, and the information from those proteins is carried out here by ribosomes, or by messenger RNA, and that messenger RNA is a copy of the DNA. So the whole goal here, the whole idea of the central dogma of DNA is, how do I get the information from the DNA molecule out here and turn it into a protein? That's what's happening here. Okay. I think I've already covered all that. Now, there are 3 players in this game, and we are going to be talking about 3 types of RNA. The first one is called messenger. We've already talked about that. That's this big long guy right here. The second one is called a ribosomal RNA. Now, we've talked about ribosomes. If you remember back in another set of videos, we talked about ribosomes, and if you remember, the ribosomal function is to make proteins. Well, this is what we're talking about here. A ribosome is really just a big RNA structure that has a job to do, and it reads this messenger RNA and puts these transfer RNAs together in order. Now, this transfer RNA is kind of the hardest one to understand because on the bottom of it, it has nucleotides, and on the top it has a protein. And this is where we're going to have to spend a lot of time talking about it. We're not going to do it here, but we got three parts here: the mRNA or messenger RNA, the rRNA or the ribosomal RNA, and the tRNA or the transfer RNA. And those are our three big players. And we're going to be talking about each one of those separately. This is involved in both processes, transcription and translation. This is only involved in translation, not transcription. Okay. And yeah, we covered all that. All right, so that's going to end this video. Go back and review this if you need to. I know it was really short, but we're just kind of setting the stage. And the next video, the next one will be video number 4. On video number 4, we're going to talk about transcription, and in video number 5, we're going to talk about translation. All right, that ends that. Go take the once you get through with this, go take the check yourself quiz to see how good you understood this, and then we'll go on to video number 4.